Very important here. Okay? And now that we see that our acetylcholine is associated with that ionotropic receptor that's going to let sodium through, is that right? If he's going to be associated with an ionotropic that's related to a sodium ion channel, that means that he's going to create with his ionotropic receptor what kind of potential? An excitatory postsynaptic potential. Very good. Understood? Acetylcholine with his ionotropic receptor, because sodium's the one rushing in, creates a what kind of postsynaptic potential? An excitatory postsynaptic. Okay? Excitatory postsynaptic. Okay? Now, I want you to see that when acetylcholine, that's what we're talking about, okay? We're talking about who? Acetylcholine. Right? He also has a metabotropic receptor. Okay? And so when he actually binds to his metabotropic receptor on that postsynaptic what? Neuron. Postsynaptic neuron. See? What's this one? The presynaptic neuron. But that's the what? Postsynaptic. So we want to look at that postsynaptic potential we can create with this metabotropic receptor, okay? And that one is going to be related to what now? He binds to the receptor, but then what's going to happen? It's a G protein coupled receptor, so you're going to have this cascade of events occur, and somewhere else on that membrane is going to be a what? An ion channel that does what? Opens. Now, in this particular case, that ion channel is not going to be what? Sodium. That ion channel in this case is going to be what? Potassium. And that channel is going to open. So wait, I'm going to ask you, right? Don't forget what we talked about before, right? If potassium's channel is open, is potassium going to rush in the cell? Is potassium going to rush in the cell? The channel's open, right? So now he can go through it, right? So he's going to go rush in the cell. Potassium rushes in the cell. You read, that's a good read. Nice, I like that. It's not going to rush in the cell. It's going to do what? It's going to do what? It's going to come out. It's going to rush out because potassium's higher concentration in the cell than out. So therefore, all atoms, all molecules have to move from what? High concentration to what? Understood? So potassium, if potassium's channel is open as a result of acetylcholine binding to the metabotropic receptor, then potassium is going to do what? And that means now, right? That means he was a positive charge, right? He was a positive charge, right? Look, the cell is already what? Look, when he binds, when does he bind? That's when the cell is at rest, right? That meant that the what? Inner membrane potential, that actual membrane potential is -70. Before he binds, it's -70, right? So when you have positive leaving, is that going to make the membrane potential less negative? Is it going to make it less negative or more negative? You got positive charge, it's already -70. Is it going to make it, go ahead and answer that. It's going to make it more what? More negative. Because positives leave, you're making it more negative. Farther away from the chance of that cell changing its polarity, right? It's going to stay that polarity that it is, right? If it makes it more negative, is the polarity changing? No, because that's already the polarity. The polarity is that it's what? Negative on the what? Negative on the inside, positive on the what? If this happens with potassium, is that going to change? No. It makes it more negative. Maybe not -70, but negative what? 90. But is the polarity there? No. So that means the cell doesn't have a what? Response. Is that right? That means the cell's not going to have a certain activity that it does. Is that right? And therefore things stay the same. So it stops things from happening. This particular potential that is created, it's a change of potential, but is it going to make the cell have a certain response? No. So it stops that process. See, this postsynaptic potential created is not excitatory, is it? It's going to stop things. So what is it? Inhibitory. Is that okay? Inhibitory. Those are the two different types of postsynaptic potentials you're going to get when you have your actual what? Messengers binding to their receptor. You're either going to have the what? The depolarization, that's excitatory, or it's going to be what? Inhibitory. Okay? So we see in this particular case, we have an example here. We see who can create the excitatory postsynaptic potential, and we see who can create the what? Excitatory. Which type of ions? Don't we see that? Yes, if the membrane potential is already starting at -70, and you have some positive rush in, that causes a depolarization. That's going to be excitatory. But if you're already at -70, yeah, and then you have some positive charges leaving the cell, then we're going to actually have what? We're going to have it being even more negative. It's going to stay at the polarity. It's going to have no cellular response. It inhibits that process. That's so. What do we have? An excitatory postsynaptic potential. Isn't that right? Right? An excitatory postsynaptic potential. And then we have what? And what? Inhibitory. Postsynaptic what? In one case, cells are active, doing something. In the other case, are they doing anything? No. They're at rest. Go take a rest. See what I'm getting to here? See, neurotransmitters have that capability. Neurotransmitters can what? Excite you. Like right now, you excite. You know what I mean? versus some can do what you feel now. How you gonna do? Yeah. But that's why people see that's a big thing. That's recreation. You might want to feel this way, or you might want to relax. Neurotransmitters. It depends on if the neurotransmitter gives an excitatory postsynaptic potential, or if it gives a what? Inhibitory. That's it. You got that? And so that's a huge thing now, okay? Related to what's going on in the world. We see our acetylcholine can do both. Okay, it depends on which type of receptors he's binding to. If he's binding to the ionotropic, excitatory, right? If he's not, if this is metabotropic, inhibitory. Got it? Okay, very good. Right. So, you know, when we're looking at that feature, okay, of excitatory and inhibitory, okay, that's really key in relationship to your different type of neurotransmitters. And this is the thing, you know, are all metabotropic receptors, are all of them related to just the potassium? So there could be some metabotropic that got the sodium too. And this is just a acetylcholine situation. I just wanted to show you what can happen in two different situations. You have ionotropic. Are all ionotropic related to just sodium? No. No. In fact, let's say I'm going to put another actual ion out here, and I want you to tell me what's going to happen, okay? Let's say the channel that opens for a certain ionotropic receptor, right? And this is what I did. Let's say it's metabotropic. I'll say it's metabotropic. Chloride. Chloride is more chloride where outside the cell than in. All right? So if if his channel's open, and what does he have? What kind of charge does he have? If his channel's open, what kind of postsynaptic potential is he going to create? Inhibitory pulse. You see that? Because it's making it more negative. The actual fibrillary potential is more negative. And if that's the case, is he making that cell do any kind of activity? No. And if that's in the brain, what's that activity going to be now? All right. They're going to go to sleep, okay? There's a certain neurotransmitter called GABA that does that, okay? GABA. He's the one that does that. Gamma amino butyric what? Acid. Gamma amino butyric what? Acid. Okay. Yeah, we got a drug. We got a drug that actually increases his activity. Is that okay? What's that drug called? Huh? What's it called? There's a drug called diazepam. Or what about this one? Benzo diazo what? No? Benzodiazepine. Nobody? That's something they give people when they come to the ER and they need to what? Calm them down. You see how that's going to work? In fact, let's say you have acidic extracellular fluid. Is that good for cells? You may pour acid on your body right now. Does that feel good? That acid, when you're acidic like that, that increases GABA. So what's going to happen to your actual brain then? What's going to happen to brain activity? See, that's that big severe case. That's what we call when your body fluid has what? Acid. When it's acidic. That's one of our main concerns. Because what are you going to do when your brain actually goes to that point and now you can't respond? What do we call that? Coma. Yeah. And that's a lot of patients in what we call the intensive care unit, right? A lot of them have what? Acidic, right? But not a lot of people, I don't see very good today. Only water. Anything else? The sodas, the juice, that makes your butt. That makes it acidic, right? So that's what, eventually after a while you start to, you know. So that's always, drink your water, drink your water. See, your neurotransmitters have control, you know. You have these as well. These guys are called the what? Biogenic what? Amines. Bio what? And, you know, in that category, we call them, within this category, there are those specific ones called catecholamines. Is that right? Now, you may have heard of some of these, right? Especially, this is the big one inside the brain. That's why we say neuro. We say nor for nervous system. We call him norepinephrine. And the one that has a high abundance where? Outside the nervous system. We call him just what? Epinephrine. You heard of epinephrine? That's a biogenic what? Amine. A biogenic amine. What does that say then? He's made of mostly what? Amino acid. That means what? Protein. Right. Okay. But amino acids, right? Amino acids, right? And so another one though is called what? Nobody heard of this one? Dopamine? Dopamine. Okay. These guys actually, you know, these guys, they're going to create more what kind of potentials? Excitatory. Okay? In